we're going to finish discussing some of the experiments people do to examine T cell development, and then we're going to move into responses in the periphery. And I will also mention, just because, that my recording on Monday failed. So I am working on a contingency plan, because many of you have told me how you use the recordings for different ways. But that one might take a little while just because I'm coming up with a backup plan. My normal way of recording didn't work for that one. So that's the story for that if you're looking for the stuff we did on Monday. It will show up, but I do need a backup plan. Today we're going to be talking about T cell responses, and one of the reasons why I want to focus here is that by and large, when we are measuring some of these interesting aspects of T cell development, the outcome that we actually look at is what kind of T cells do we see in the periphery. So we're going to, in some cases, be looking at T cells in the periphery as our outcome to tell us what happened during T cell development and selection. I just want to remind you, because this will really clarify some of the experiments that we're going to talk about, that throughout T cell selection and development, our developing T cells, which are these orangey red cells in this figure, are making many interactions with cells of the thymus. So in the cortex, our cells are interacting with cortical thymic epithelial cells to test against antigen. A lot of the positive selection processes are happening here. So if we were not able to interact with that cortical epithelial cell, the cell would not survive. Our cell would die by neglect or would fail positive selection. You can see that similar interactions happen in the medulla, where medullary thymic epithelial cells are presenting antigen for testing for negative selection. And those medullary thymic epithelial cells are the ones that are producing air, so we can get the wide variety of antigens presented. Someone was asking me about this after class on Monday. Air is largely expressed by medullary thymic epithelial cells, so they are the ones that are translating kind of all the proteins in the body. It's not the entire thymus makes every single protein in the body, because then we start to get into like the existential crisis of the thymus and what makes the thymus a thymus. But it's really just the MTECs that are actually doing all of this broad transcription and translation. Earlier on in this process, those developing cells also needed to interact with cells of the thymic stroma, the structural cells of the thymus, in order to acquire cytokines to help support survival. The reason why I reminded you about all of these interactions with thymic cells is because this is the reason that T cells are always restricted by the MHC of the thymus. So as I told you guys last time, if we perform radiation on a mouse or a person, the cells of the thymus are not altered. And thus, even if we do radiation in a bone marrow transplant, our mouse, let's say, will end up with T cells that are still restricted by those thymus cells. And that's because those thymus cells are really important for presenting antigen. They're not affected by the radiation. They are usually referred to as radio-resistant cells. And so here you can see a couple situations where we have a yellow mouse and a blue mouse who each receive radiation. And they each receive a bone marrow transplant from an orange mouse after that radiation. So notice that the bone marrow cells start out with the orange genetic background either way. When those cells are transplanted into the yellow mouse, they end up making responses to yellow MHC type. When we have that response going into a blue mouse, we end up with a response to blue MHC type. So these cells basically are always educated that self is the type of self that is in the thymus. because of those important interactions with thymic epithelial cells. And so even though all of these bone marrow cells started out as orange, in the end, the thing they recognize, the thing they think is self, will be whatever was the MHC type of the thymus where they are put. You can see that also in this figure from your book. It's really the same type of experiment. So here we can see a mouse who has both type A and type B MHC. So it's an F1 of type A and type B. So it should have a chromosome of A types of MHC from mom and a chromosome of B types MHC from dad, or vice versa. We can infect with a virus like LCMV, and T cells from this mouse that came from the spleen will be able to kill strain A infected cells and strain B infected cells. Though there will be some T cell that grew up in this thymus and saw A as self. There will be a different T cell in this mouse that grew up and saw B as self. It's a question of which MHC type in the thymus did that cell interact with. It's not that every cell is going to recognize like A and B combo, but some are going to learn A, some are going to learn B, depending on which MHC they interact with in the thymus. And then they will be able to make these responses. So if you take all the cells together from the spleen, some of them will recognize A, some of them will recognize B. If we then took that same mouse and removed its thymus and did radiation, so here you can see the thymus has been removed. Why it is the shape of a spleen, I don't know. And we put in a thymus graft, a transplant, that is just strain B, just H2B. Even if we put in the AB stem cells, the resulting T cells are all going to be restricted by B. We're not going to have any A-restricted cells because every cell that grew up in this thymus learned B as self. Yep, Emilio.
So is every T cell restricted to a single MHC?
Every T cell has a T cell receptor that is specific. We have allelic exclusion, so each T cell is making one T cell receptor. And that T cell receptor will bind a particular type of MHC plus peptide well. We can talk at some point about cross-reactivity because cross-reactivity is a thing, although I don't want to get crazy into it now. But cross-reactivity theoretically is possible, so I'm not going to say it's 100% always only the one kind. But in theory, each T cell has one peptide MHC pair that it binds to, and that includes a specific MHC type. And so you can see this experiment happening here. This also can lead to a few different types of other experiments. I don't like the placement of the next slide, so I'm going to skip it for right now. We can do experiments where we have mice who are missing either MHC class 1 or MHC class 2. Because having your thymus present antigens on class 1 or class 2 is very important to allow those T cells to develop. If our mice are missing class 1 or class 2, then that will impact what kind of cells we find in the thymus and also what kinds of cells we find in the periphery. So control mice have double negatives, which are the earliest cells in the thymus. They have double positives, and they have both kinds of single positives. If cells are deficient in class 1, we still have double negative cells. Those cells still start developing. Though we still have double positive cells, those cells still continue developing to the double positive stage. But when those cells get to the point where they're trying to become either CD4s or CD8s, they're not going to be able to become CD8s because there is no ligand that goes with CD8 class 1. So in a class 1 deficient mouse, you end up with no mature CD8 positive T cells. You also see no single positive CD8 positive T cells in the thymus. You still get the double positives because, in fact, double positives are made earlier during development. If we make a class 2 deficient mouse, Same thing, we still have double negatives because we have those early developing cells. We still have double positives because we have those cells that are trying to develop. CD8s are still going to be fine because they're going to interact with class 1. However, we're not going to make any CD4s in this mouse because no cells are going to be able to develop along with MHC class 2. And so these mice absolutely can be made to work in the laboratory. We can also, and we very frequently also, make mice that are known as TCR transgenic mice. These mice are actually quite similar conceptually to some of the B-cell transgenic mice we saw earlier in the semester. But for some reason, people get a lot more confused about these guys than they did with the B-cell mice. So the idea here is that we take a mouse, like this H2K mouse up at the top, and we isolate some T cell from that mouse. Here you can see the T cell that we have isolated from that mouse. That T cell will respond to some MHC from that mouse. Here we tell you that this mouse is of an H2K haplotype. We can take a CD8 T cell from that mouse. We know that CD8 T cell must recognize some antigen that is presented by class 1. It's presented by some molecule. It could be K of K, for example. We don't know. Let's pretend it's K of K. And it might be, say, the flu T cell. We would pick the flu T cell out of that mouse. Here we are. We picked it out of the mouse with an arrow. And you can see that it would normally recognize a flu-infected cell. We could then take that T cell and isolate the genes encoding the TCR alpha chain and the TCR beta chain from that cell. These will be already rearranged. So we can take the rearranged alpha that this cell made. We can take the rearranged beta that this cell made. And then we can take these genes and place them in the genome of another mouse transgenically. So we can insert those pre-rearranged alpha and pre-rearranged beta chains into the genome of another mouse. We saw this exact phenomenon with B cells. And so what will happen is that as all of our T cells develop, this alpha chain and this beta chain will be made immediately. They will allelically exclude other alpha chains and beta chains. And so 100% of the T cells in this mouse will recognize this influenza epitope on K of K, and will basically all be versions of that one T cell. 100% of the T cells in that mouse will become this T cell. And so because we chose a CD8 T cell at the beginning, and because we chose a T cell that recognizes class 1 at the beginning, only CD8 T cells will be found in the periphery of this mouse. There will be no CD4 T cells in the periphery of this mouse. If we look at the thymus of this mouse, we'll see double negatives, as we would expect, because those cells are starting development. We'll see double positives, as we would expect, because those cells are doing development. But then all of our single positives will be CD8s, and there will be no CD4 single positives. Yeah, Molly?
What's the experimental advantage of this?
So that's a great question. Well, Molly asked the experimental advantage of this, and that's exactly the question you should be asking here. So first I'm going to tell you the experimental disadvantage of this. So let's imagine that we make this mouse, and this mouse escapes the mouse facility. So I want you to remember that immunologists tend to do their experiments in mouse facilities that are called specific pathogen-free, that are very, very clean and have no pathogens in them. Our mouse facility back here isn't fully up to SPF. So let's imagine we had a mouse, we made this mouse, and it escaped out into the real world. You can imagine it being the mouse that we had a mouse recently escape our mouse facility, and it got into Dr. Dunaway's desk and ate his gum. And he was very upset about it eating his gum. You can imagine that happening. And that happening to this mouse that we've just made with these T cells. How do you think life would go for that mouse if it escaped into the real world of Dr. Noe's gum? Huh?
It would die
It died pretty quick. Why?
because it only has defense against.
Yeah, it would be amazing at getting rid of influenza. If it happened to encounter influenza, woe for that influenza. But it couldn't address any other pathogens. And so in terms of thinking about physiology of these mice, they're not super useful. But they are super useful for two experimental purposes. One of them is just simply doing experiments on T cell development. I'm going to tell you a little bit about a really famous set of experiments. In the past, we've read this paper, but for a bunch of reasons, we're not going to read the paper this year. And they were trying to determine things like, does deletion happen to T cells, as we saw with B cells? And they had the problem of, if you have a rare cell and it gets deleted, how do you prove it got deleted and not just lost? And that you couldn't find it because it's a rare cell. And so what they had to do was come up with ways that they could make the cells be really, really frequent instead of really rare, so that they could track a whole population instead of trying to find needles in a haystack. When 100% of the cells recognize your antigen of interest, it's pretty easy to see what happens to them. The other thing that can actually be really helpful, and the reason why people do this pretty frequently, is these can also be nice sources of cells. So sometimes you might want to get some influenza-specific cells to, say, transfer into another mouse. Immunologists really love to do transfer experiments where you transfer cells between organisms. This is an awesome source of cells. And so these mice are sometimes made as cell sources as well. Yep?
Might be a dumb question, but would the hypothetically escaped mouse die anyway because of autoimmunity? Like due to the strength of response to a singular pathogen? Because an influenza pathogen would turn on every single T cell in its body. That would then proliferate a lot. So wouldn't it die anyway?
So Molly's question is, well, what happens if this mouse, it escapes and it does see influenza? Molly says, wouldn't it die anyway because it would actually have too vigorous of an immune response to that influenza? It would have some kind of immunopathology. The answer is maybe. I'm not sure if anybody has actually, I'll have to look and see if somebody's done that experiment. The other issue is that CD8 cells need CD4s to help them function, and this mouse doesn't have any CD4s. So I'm not actually sure if that would work. I don't remember off the top of my head if that experiment has been done. But so the mouse would be in bad shape, kind of no matter what. We really do not want this mouse to escape and run free. I mean, that would be nice for the mouse, I suppose, but then it would be really not nice for the mouse. But these types of experiments are done quite frequently. So if we were to do an experiment where we give the cells, or give the mouse, the alpha and beta chain for a transgenic receptor recognizing class 1, only CD8 T cells will mature. If we give the mouse an alpha and a beta chain for a transgenic receptor recognizing class 2, then only CD4 T cells will mature. So that's one way that people do these types of experiments. But people use these experiments in some other ways as well. So this is one experiment that you could imagine. Someone did do this experiment. So we can take our mouse, that is an H2K mouse. We can clone out the alpha and beta TCR genes from one of its T cells, and then we can insert them as transgenes into new mice. We could put them as transgenes into mice of the H2K MHC type, which, as you notice, is the same as the original. We could also put them in a mouse of the H2D type, which is different than the original. If we put them into the mouse that's the same as the original, well, we get lots of developing thymocytes. In this figure, we just see double positives here to tell us about developing ones. There would be double negatives too. We'd get lots of cells that are developing. And this whole experiment would work. We'd get 100% of this mouse's cells would be CD8 positive T cells. as we hoped. If we, however, did the experiment in this way, we would get a different outcome. We would still see cells that are trying to develop. We would still see the double positive cells. Except when those cells tried to actually see their correct MHC, the MHC that they are specific for, which is K, perhaps it's K of K, they would look for their MHC and they would never find it. Because this mouse is an H2D mouse, not an H2K mouse. There would be no MHCs that matched these T cells. And so all of these T cells would die of neglect. Every single one of them would not be able to see MHC. And so in fact, we would get 0 CD8 positive T cells. We'd also get 0 CD4 positive T cells. We'd have no mature T cells in this mouse. And so this is actually one of the ways that we really started to understand the fact that T cell development did require the MHC type. And this is what was covered in that slide I skipped before because I don't know why I decided to put it in this weird place. So if we put in a transgenic receptor that is restricted to MHC type A, and our cells of the thymus, our thymic stroma, also expresses that same MHC type, our developing T cells can interact with that stroma and undergo development so that our single positive cells do in fact mature. If we put in a transgenic receptor restricted to MHC type A again, however, if those cells are in a thymus of MHC type B, so that the stroma is MHC type B, the cells will try to interact, but they won't get anything. They won't get any signal. And so all those cells will die. Sad apoptotic cells. And in the end, no single positive cells will mature, and this mouse will have no T cells going forward. So this was how some of the aspects of positive selection were first described. Then an additional experiment was done that helped us understand both positive and negative selection. That experiment is shown here. In this experiment, again, we took a T cell out of a mouse. This T cell recognizes an antigen on the MHC type H2D of B. So you can see it says H2D of B restricted at the top. So that's the MHC type here that is the right MHC type, is D of B. And this T cell recognizes a specific antigen, which is called HY. HY is a protein that is encoded by the Y chromosome. So in males, this is a self protein. And in females, this is a non-self protein. So they found these T cells. They basically made T cells. They got the alpha and the beta chains from them that could recognize an HY peptide on H2D of B. In female mice, these are going to be perfectly good normal T cells that recognize some foreign antigen. In male mice, these are going to be T cells that are autoreactive and lead to autoreactivity. And so these experimenters realized that with this system, they could actually see how autoreactive T cells were affected. They could use the transgenic mice so that 100% of the T cells should be HY specific, and they could actually see, like, are we really having the cells get deleted, or is it just that it's hard to see them? So they dealt with a needle in a haystack problem, and they could get around anybody who wanted to argue, you used a super weird antigen. They could show that the T cells developed correctly in females, because they're non-self in females, and then they could show what happened to them when they were self in males. So they did this really nice controlled experiment. The reason why this is a painful paper to read, it's a beautiful paper, but it's really tough to read. And the reason this paper was only possible because they figured out how to make these types of transgenic mice, which was a very new technique at the time, they also had the ability to do flow cytometry, which was a very new technique at the time. So they were able to do both of them. The flow cytometry looks a little different than some of the stuff you're used to. But there was a thing they didn't know when they did these experiments. The thing they didn't know is about TCR alpha, and the fact that TCR alpha is terrible at allelic exclusion. So when you do this experiment, while I told you before 100% of your T cells become the HY T cell or the flu T cell or whatever, it's actually not true because TCR alpha can't allelically exclude very well. And so when you do these experiments now, you actually also knock out RAG1 and RAG2 in the mouse. So not only do they have to use this alpha and beta chain because you put it in and it rearranges, but even if the cell tries to rearrange, it can't because it has no RAG1 and RAG2. So that's the way you do it now, and you do get 100% HY cells, or you do get 100% influenza-specific cells. They didn't know that at the time, and so the data is really messy, and it becomes difficult sometimes for students to totally interpret the data because there's some weird things going on because of that lack of allelic exclusion. So we're going to kind of talk about big picture what happened. Some of the figures from your textbook, I feel like they've kind of cleaned them up to look a little bit more like what we think the experiment would look like. So we can first take these cells and we can put them into female mice of who have H2D of B. These mice don't have HY expression because they're female. They make some double negatives, they make a lot of double positives, and they make a lot of CD8 T cells because, in fact, we put in a CTL clone. Here it shows they make a few CD4s. Don't worry about those. And so realistically, what we see here is sort of what we would expect. Yeah, Marina.
I'm sorry, but like for the one on the right,
Mm-hmm.
why is it 2 negatives for CD4 and
Yeah, so that one I haven't gone through yet. Good question. So if we look at a wild type mouse that came from the mouse store, and we look at its thymus, we would expect to see something that looks like this, where we'd have a few double negatives, a lot of double positives. We'd have some single positive CD8s and some more single positive CD4s. So this is what we would expect to see with a wild type mouse. If we look at our mouse who is the female H2 that has H2D of B, so the one in the middle, we're not at the one that Marina has a question about yet. We see some double negatives, we see a lot of double positives, and we see some CD8s. We don't really see CD4s because we put in a CD8 T cell. We should get CD8 T cells out. And so this is what we see in this mouse. We can then do the same experiment in a mouse of a different a female mouse of a different MHC type. So this mouse is an H2D of D mouse, not a D of B. This mouse does not have that type of MHC. And so here we see some double negatives and some double positives the cells are trying to develop, but we don't see our CD8 single positive cells. These cells are missing because the cells are not able to develop. These cells are all going to die by neglect. They test their receptor against the thymic stroma. The thymic stroma doesn't bind to them, and they all die by neglect. And so in our D of D mouse, and again, we're still in females, we see double negatives, we see double positives, and we see no single positives. And so, in fact, here are the data. So you can see that the D of B mouse data is here. Yes, there's this extra little group of CD4s. That's part of that messiness with allelic exclusion. We can ignore those. You can see, if nothing else, that there is a dramatic increase in the CD8 population. If we have a mouse that is the progeny of a B and a D mouse, then in fact it also nicely makes CD8s because its thymus does have some cells that can support differentiation of these developing T cells, so we're good. If we use a female H2D of D mouse, we can't get any single positives because we're unable to support the development of those cells. And so that was kind of the first big thing that they did. But as you might guess, part of the point of doing this experiment is to also look at the development of the T cells in males when these are now autoreactive T cells, which they were not previously in the female mice. And so they did this same experiment where they put these alpha and beta TCR genes in an H2D of B mouse. This is a male mouse who does express HY. So now this is a self-antigen. And they see something slightly different here. So first of all, I want you to look at the one on the right. This is our female H2D of B mouse. This is our female mouse where we have put those developing cells in the correct MHC type. We would normally think it would look like this with no CD4s. We do have that little messy CD4 background. This is those same cells in a male mouse. You can see that, yeah, you get some cells up here, but they're not like in the right place. There are some cells that happen to fall into the double positive gate, but they're not real double positives. Like if you look at what actual double positives look like, that's what double positives look like. This is just messiness. And similarly, these aren't real single positives either. They just sort of messily end up there. Really, in this mouse, all we get are the double negative cells. And you can see that we get a lot of double negative cells, way more than we get in our other types. Because in fact, these cells are blocked at the double negative stage. As soon as they are unable to progress to double positive. They're unable to get any signals through this. And once they do actually get to the double positive stage, they are actively killed because they are self-reactive. And so it's not that these cells are developing and trying and just sort of hanging around until they undergo apoptosis. As soon as these cells are made as double positives, we kill them through negative selection and we get rid of these cells. And so this is sort of what we end up with in these mice. And so this is a very famous experiment. This is actually how we learned that developing T cells start out as double negatives and then become double positives and then become single positives. That timing and order was learned from these experiments. The ideas of both positive and negative selection were learned from these experiments. A lot was learned from these experiments. They were done by a really interesting investigator named Harold von Boehmer, who actually passed away earlier this month. So at this point, our cells are done in the thymus. And our B cells that we made before are done in the bone marrow. And so now we're actually ready to have them make some immune responses in the periphery. So these are two different figures of cells moving to the periphery from your textbook. You can see here that we had some B cells that we developed in the bone marrow. We've got our CD4 and CD8, or helper and cytotoxic T cells that developed in the thymus. And now they are going to leave and go to all of these wonderful periphery places. And by and large, they're going to start their responses and really start dealing with antigen by making some responses in the secondary lymphoid organ, as is shown here. And what you might notice here, and you'll see this again later, is that there's a problem of travel that has to happen for these cells. These cells now have to actually move from their developmental site to the site where they can find antigen, or to the site where an infection is occurring. And so to start to understand the process of making an immune response to an antigen, we have to think about how the cells deal with this traffic problem and how the cells start to understand their location. I think these two figures sum up a lot of things for me. They crack me up a little bit. These two figures address the question of where are lymphocytes. So first I'm going to show you the one on the left, which is from an older version of your textbook. One thing that it does, that it tells you, which is correct, is that your cell, any lymphocyte is able to make a full circuit of the blood. So if it so from heart, out of the heart, around the body, back to the heart, it can make the full circuit of the circulatory system in 30 minutes. So some portion of our lymphocytes are making this circuit every 30 minutes. However, some of them leave the blood and go to mucosal tissues. Some leave and go to the spleen. They might stay there for 5 hours. Some might go to a lymph node. Some might go to the lymphatic system. And so our cells are frequently found in some of these other places. You can see that here as well. The cells will leave the bone marrow and the thymus. They will end up in the blood flow, which is depicted here by the heart. And then they will travel around, potentially to tissues like the skin or the GI tract, potentially to the spleen, potentially to a lymph node, through the blood, into all of those different places. The thing that cracks me up about these two figures are the numbers. If you add up the percentages in the figure on the left, what do you notice? It's way more than 100. And if you look at the answers between the two versions of the textbooks, they don't exactly agree either. And in fact, this is sort of a question that we have a hard time answering in immunology, is how many of the lymphocytes are in what place. It's relatively easy to draw some blood and to count the number of lymphocytes and say there are this many lymphocytes per microliter of blood. It's really hard to take all of your skin and digest it with collagenase and pull all the lymphocytes out, which is actually what you have to do, and then count it and then figure out, like, what is the units? Is it per this many grams of skin? Is it per this many centimeters of skin? So in fact, doing these types of experiments is quite challenging. Coming up with real numbers here is quite challenging. You will find, if you watch TV and you see some of the medications, particularly like probiotics for your microbiome, there is one commercial, I can't place it now, that says 90% of your immune system is in your GI tract. And that's actually because there are papers that indicate a much higher percent of lymphocytes living in the epithelial wall of your GI tract. And people argue what that number is all the time. You know, it could be like between 5 and 95%. So, you know, not too much of a range there. So these numbers are very unclear. These questions are actually quite challenging to study. But we're going to think a little bit about how we study these problems, and more importantly, how the lymphocytes actually make these movements and how they go to each of these different locations. They don't have a car. We're going to talk a lot about cars with this. They don't have a car. They also don't have a brain to read street signs. So how does a lymphocyte know I'm in the blood, I'm in the spleen, I'm in the lymph node, I'm in the skin, to potentially make those responses? How does it know where it wants to go? And how does it get there? And how does it know when it is there? And so all of those are incredibly important parts of this. We're going to be seeing a lot of activities occurring in a secondary lymphoid organ. I'm usually going to use a lymph node as my example secondary lymphoid organ, though most of them have pretty similar structures. What you should notice for your secondary lymphoid organ is that we have a blood vessel in, we have a blood vessel out. So we've got blood in and out, and you remember you saw that when we dissected the mouse and I showed you a lymph node, and I told you that most immunologists, when they do dissections, find lymph nodes by looking at places where vessels interact that look like snot. Because there's that connective tissue right around it where the vessels are interacting. We also have a lymphatic vessel coming in, so an afferent lymphatic, and we have the lymphatic vessel coming out, an efferent lymphatic. Some of these blood vessels are going to be really specialized, which we'll talk about a little bit more. And within that lymph node, there are a lot of specialized areas. There are areas that you can see here listed as cortex and medulla, follicle and paracortex. The thing that I want you to realize is that there are specific areas where the B cells are, specific areas where the T cells are. So the cells are actually quite organized within this lymph node. And so that can be another trafficking problem. How the heck does this cell know where to go in the lymph node once it gets to the lymph node? All of these issues are part of a bigger problem with the adaptive immune response. On the upper left of this slide, you can see a cartoon version of antibody and T cell responses following a first infection. And what we talked about way back at the beginning was that the sort of downside of the adaptive immune response is that it's slow, that it takes a while in order to make a response. And you can see that we start to get a really high antibody response, a little more than a week after our infection. We start to get a good T cell response a little more than a week after our infection. People always hate the cartoon versions of this figure because people look at this and they're like, Dr. Barker, no, I want a number. I want a day. I want it perfectly written out. So I'm giving you one sample graph from my PhD where you can actually see the days. Just look at the black solid line. You don't need to look at the dotted line and the open things. So here I inoculated mice with adenovirus. I was really able to start measuring my CD8 T cells at day 7. They peaked at day 17, and I was able to measure them for the 8 weeks of the experiment. The actual reason why you never see this with numbers is because the specific numbers vary a lot based on the pathogen and the dose and the route of infection. And so usually it's hard for us to give you I can't tell you your adaptive immune system is always good on day 12. There isn't always a specific day because it varies based on the type of pathogen. I did this identical experiment with a different pathogen and my peak was at day 10. That varies, but if you want numbers, there's a version with numbers. And you might wonder, well, what the heck takes so long? What's the adaptive immune system doing that's taking such a long time? In reality, there are a bunch of answers here. One answer is that we need to have things like production of antibodies and effector T cells. We need to have proliferation of cells. We need to have maturation of cells. So those are big parts of this, and those are parts that we're going to see later in the process. But one of the things that takes such a long time is transportation, is getting the antigen to the secondary lymphoid organ, or getting the T cell to the secondary lymphoid organ, or getting the T cell back to the place where you're infected. It's great if you have a whole bunch of T cells ready to kill chickenpox in the lymph node, but until they get to the skin, your rash is not getting better. And so travel to the lymph node and travel from the lymph node are one of the big parts that take so long. And so for many of our next few class periods, we're going to be seeing reasons why the adaptive immune system takes this long of a time to get going. And so this trafficking issue is, in fact, the first of the reasons why it takes so long. You can see kind of in general some times listed for T cells and B cells. So you can see that, for example, antigen is going to get into the lymph node usually within minutes after infection. But the B cells are probably going to take a couple days, maybe up to a day to get there and to start actually proliferating. They're going to take a couple of 2 to 4 more days to develop. So this is sort of what's going on. Similarly for T cells, it's going to take a while. With T cells, we've got to get an antigen presenting cell, like a dendritic cell there. We've got to get antigen there, and we've got to get the T cell there. And so we might get the APC there quickly, we might get the antigen there quickly, but then we've got to spend some time waiting to get the T cell there, and we've got to get all 3 of those together, and then the T cell has to do some work. So you can see that there are a lot of events that are going to happen that are going to tell us why it takes so long. And overall, we are going to care about how did the antigen get there. We're going to care about how does the B cell get there, although we're not going to be focusing on B cells so much today. We're going to care about how does the antigen presenting cell, like the dendritic cell, get there. And we're going to care how does the T cell get there, all to get to that lymph node. You might guess from this figure that the antigen gets there pretty easily, because it says the antigen gets there in minutes. So that must not be very hard. And in fact, antigen transit to lymph nodes is really not a big deal. Here is a lovely example of a person stepping on a rusty nail. You can see that person stepping on the rusty nail here. There are likely little red dot antigens. And there are open lymphatics throughout the body. Remember I told you about the lymphatic system, how it's this open system to collect water, to collect the interstitial fluid that has leaked out of the circulatory system. So because we have this open system of vessels, not only can liquids flow in, but any sort of particulate antigens, even things like bacteria, can also just get swept into the movement of that fluid. And so the antigen is basically just going to move through the lymph pretty easily. In most places, there are some antigen-presenting cells, like immature dendritic cells that are living in that area, that may be able to also pick up the antigen and move it. But if they can't, the antigen can just move perfectly well by itself. And so here you can see we have a wound in the skin. We've got these open vessels, lymphatic vessels, and we can have bacteria, we can have antigen, or we can have APCs carrying that antigen. Just go through the lymphatics, and eventually they will hit the nearest lymph node for that region of the body, the draining lymph node. Here are some examples of draining lymph nodes. So basically, they will just get swept up and move to the nearest kind of local lymph node that drains that area of the body in order to start a response. And so as I told you guys before, a lot of times when you have a sinus infection, you get swelling in these lymph nodes. It's because these are the lymph nodes that drain your sinus cavity. You've got the lymph node behind your knee that's draining your lower leg. You've got a lymph node in the groin that's draining your upper leg, and many of others throughout the body. So we can get the antigen to a draining lymph node really easily. That's not a big problem. The bigger problem is getting the T cells and or B cells there. T cells have two ways they could get to the lymphatic organ, to our lymph node. Our T cells can travel through the lymphatic vessels, so we can have travel through the lymph. Our T cells also can travel through the blood and make the decision or the choice, and all of those are anthropomorphizing T cells in a way that is not true. But our T cell can then leave the blood vessel and enter the lymph node, or that cell can be just moving through the lymph. So both of those are possible. And this general problem is not specific to this part of immunology. So in fact, if we want to see a T cell or B cell come into a lymph node, it has to go through this trafficking process to go into the lymph node. If we have a memory T cell or an activated T cell that wants to go to your skin to kill the chickenpox rash, it has to figure out a way to get out of the blood vessel at your skin in order to kill the rash. Here, our T cell had to figure out a way to get out of the blood vessel into the lymph node. And there are lots of situations where your neutrophil might have to get out of the blood vessel in order to go to a site of infection. You saw that at the very beginning. It also might be a local macrophage like this. So this process of how does a white blood cell leave the blood vessel, how does it say, ha, this is my spot, and then leave the blood vessel, is actually a common problem through most types of immune responses. We could have gone through this whole thing when we did inflammation and the neutrophil response, but we're instead going to go through it all now. It would be exactly the same problem if we had done it with the neutrophil. There have been some really cool technological developments that have allowed us to study this process. You're going to see some data from one of these today, and you'll see some other data from other such experiments later on in later figures. And this is actually using a technique called intravital microscopy. So intra is in. Vital means alive. So in fact, in intravital microscopy, you are doing microscopy and watching cells move in a live animal. Because if you think about it, it's kind of hard to measure these trafficking problems, how the cells navigate the body in a dish. because you need a body to measure them. And so people actually came up with a way to do intravital microscopy and actually watch the cells move in a living animal. Very frequently, we will insert T cells or dendritic cells of interest that are fluorescently labeled because it makes them easier to see under the microscope, though you actually don't have to do that. The first video I'm going to show you today, they're not labeled. You then take your mouse and you want it alive, but you're going to put it under anesthesia because a mouse is not going to just lay on your microscope slide by itself. So you're going to anesthetize that mouse, and you are going to perform surgery so that you actually can expose its lymph node. You do this lymph node basically with the lymph node at the groin because that's the one that's closest to the skin, and it is not really very deep. So you surgically make it so you can actually access that lymph node, and you take your anesthetized mouse and keep it in place with the lymph node open. You can then use a microscope to actually look directly into that lymph node and watch the cells move. Very typically, this is done with confocal or some of the interesting microscopy techniques that Dr. Larson's lab does. So here you can actually see the node coming out of the mouse that you can actually check pretty easily. This was an idea of a mouse being restrained. Usually you're going to have maybe a pump of anesthesia, either nasally or through the tail vein. Usually we'll have a heat circulator to keep the mouse warm. And you'll be able to just actively do microscopy on that open lymph node. And so this has been a big way that they have been able to study and understand a lot of parts of this process. And there are a bunch of problems here. So we've got to think about what the problems are before we understand what the solutions that the immune system has smartly come up with are. So here again is our immunologist view of the circulatory system. We talked about this with pressure and why we needed to have leaky vessels and why we got interstitial fluid before. And we talked about how at the beginning we have this big pump pushing things. There's a lot of pressure on the cells. And this can become a bit of a problem for white blood cell trafficking. I want you to think about why this is a problem for white blood cell trafficking, but I'm going to show you an image, a video with an intravital microscopy image to help us think about why this is a problem. So the image I want is this one. So here you are able to see white blood cells moving through vessels. Here you can see some slower moving cells. You can see kind of the entire aspect of the mouse here. This is not fluorescently labeled, obviously. This is just active cells. And you can look at those cells moving. What do you notice, or what might this remind you of? And I want you to think carefully about what is the diameter of those cells. That might help. Yeah, Molly. It made you think of highways. That's awesome because my metaphor for this whole thing is highways. So what does it make you think about with highways? Oh, no, not ads. No, I don't. I do not want to advertise grammar. Blood flow. And in fact, in this, you actually can see the mouse move its leg. In this, reminds you of highways. What does it make you think? What kind of aspect of highways does it make you think about? Or what's the problem here with blood flow? What?
You can't get into the left lane.
There's this question of how do you get into different lanes? So I'm going to drive to my parents' house for my 20-year high school reunion this weekend, so I'm going to be on a highway. Am I going to drive for 5 hours on the highway and then go and keep going at 80 miles an hour the whole way? No, why not? What would happen if I did that? I would crash. Why would I crash? What would happen to me? I'd be going too fast. I might, if the people next to me are going a different speed than me and they ran into me, that would kind of shear me in half, right? Yeah, and in fact, if a blood cell, if you actually think about what the diameter of those cells are relative to that speed, this is incredibly fast movement for these cells. If this cell got to, aha, here's the skin, I am going to turn and go through the blood vessel wall, these cells would actually be sheared in half by their neighbors that are moving so quickly around them. And so, whoa. All right, so if a cell actually wants to leave the blood flow, whether it's a neutrophil wanting to go to a site of inflammation, whether it's a T cell wanting to go to the lymph node, whether it's a T cell wanting to go to a site of infection, that cell is going to have to do things like you do on the highway, because it's going to have a lot of the possible problems that you would have on the highway. So what do you have to do on the highway? Tell me what I should do on the highway when I drive to my parents' house this weekend. So I'm going to need to have a signal, right? Okay. So I'm going to signal. What else might I do? Slow down. What else might I do? So I might slow down. But, huh? What? I'm probably not going to honk. Oh, only because cells can't really honk. It would be hard for us to make the cells honk. What else do I have to do? I'm driving super fast down the highway in upstate New York. Driving, and there's my exit over there. What do I have to do? Slow down. I have to slow down and signal, but there's one other thing I have to do. Huh? What do I have to do, Molly? Yeah, I have to get in the other lane. I have to get in the slow lane, out of the fast lane. And in fact, the cells are basically doing that same process. Your cells are able to leave the blood vessel via a 4-step process. One of these steps is much less understood than the others, so really I'm going to focus a lot on the 3 steps, and the 4th step is not going to be understood. These 4 steps are shown here from figure 14.4 of your textbook. The big 3 are rolling. Rolling is also sometimes known as sticking. Then we have one that's listed as activation. We can also think about activation as being something like signaling or chemokines. You'll see. The third one is here listed as arrest or adhesion. Sometimes you will hear it referred to as firm arrest. Sometimes you will hear it referred to as stopping. And then our cell is going to undergo this 4th step, which is the one I focus on the least, that's listed here as either diapedesis. Sometimes it's called extravasation. Sometimes it's called transendothelial migration. So diapedesis is on the slide, so hopefully you can spell that one. But that's extravasation. Or transendothelial migration. So it's really the same process that I'm going to follow on the highway. So if we think about this for a second, about my highway trip, we're going to continue with my highway trip on Friday, which is great, because then after we do that, I'm going to go on my highway trip. So I'm on my highway trip. What's the first thing I need to do when I get to my exit? Huh? I probably want to slow down. I also need to recognize that it's my exit. So I need to recognize this is my exit, and I need to start to slow down. And the first step, rolling, is the step where the cells start to slow down. These cells start to recognize this is a place where they might want to leave the vessel, and they might want to slow down. This is another video I'm going to show you of rolling. Just so you know, if you go to this website, it doesn't work, which I learned right before class today. It's from a textbook. Fortunately, everything from that textbook is on YouTube, so I found it on YouTube. But if you go to this link, it won't work. I searched Garland rolling, and I got it. And so here are our cells doing rolling.
Leukocytes are white blood cells that help fight infection. At sites of injury, infection, or inflammation, cytokines are released and stimulate endothelial cells that line adjacent blood vessels.
I really just want you to see this
The
step.
endothelial cells then express surface proteins called selectins. Selectins bind to carbohydrates displayed on the membrane of the leukocytes, causing them to stick to the walls of the blood vessels. This binding interaction is of sufficiently low affinity that the leukocytes can literally roll along the vessel walls in search for points to exit the vessel. There, they adhere tightly and squeeze between endothelial cells without disrupting the vessel walls, then crawl out of the blood vessel into the adjacent connective tissue.
So here we're focusing on that
Here,
rolling step.
Leukocyte rolling is observed directly in an anesthetized mouse.
Look at how fast all the rest of the cells are
The
moving.
up-and-down movement of
And this is the rolling cell.
mouse is breathing.
So we really slowed that cell down.
The upper one is an artery with blood flowing from right to left. The lower one is a vein with blood flowing from left to right.
So you can see that this rolling step slows that cell down substantially and is really going to help that cell not get sheared by its neighbors. You'll also see that it does pull that cell to the side. It pulls it over into the exit lane, to the side of the wall, as well as slowing it down. This is happening with interactions with a particular type of protein called a selectin, which I'll start with on Friday. But what I will just tell you right now is when I think of selectins and selectin affinity, I think of Velcro. They come on and off really, really, really easily. And so the idea with selectins is that they're catching and releasing and catching and releasing and just slowing the cell down and pulling it over, but not making it stop to a point where it's going to get sheared. So we'll finish up with this information on Friday. I'll see you guys then. Please bring your drafts for lab tomorrow.